This week we start our assignment talking about how cells divide. In eukaryotes, we're going to focus on cells dividing in eukaryotes and not in prokaryotes. In prokaryotes, it's fairly straightforward. Remember that all eukaryotes contain a nucleus and membrane-bound organelles. That means things like the mitochondria have a membrane around them. All eukaryotes go through some form of asexual reproduction, which simply means the cells divide by mitosis, and they do that for three reasons. To repair damaged cells, so if you get a bruise on part of your body, those cells have been damaged and need to be replaced. So the new healthy cells underneath will go through rapid mitosis in order to repair the ones that have been broken. Eukaryotes also go through mitosis for the purpose of growth. You weren't as big as you were when you were an infant. When you were an infant, you were a certain size, and your cells divided and divided and divided and divided until you grew. And lastly, for the development of the organism, your cells did not perform as specific of jobs as they do now. When your cells divide, they start to specialize, and that helps with development. So the part of the cell cycle that we're focusing on happens after interphase. Interphase is where there's not a lot going on with the cell. It's just doing its normal job. All the parts of the cell are functioning correctly, but it's not actively going through division. We're focusing on the M phase, which is M for mitosis. And if you look at a cell cycle, the cell goes through this circular loop over and over and over and over again, and this is the part we're going to focus on, this prophase, metaphase, anaphase, telophase. So let's move over to mitosis and get started. Mitosis, again, is cell reproduction and division. There are 4 mitotic stages. The first is called prophase, next is metaphase, then anaphase, then telophase. And here you can see those 4 stages. This one is prophase, this one is metaphase, anaphase, and telophase. This is a very close-up microscopic picture of the prophase stage of mitosis. You can see the blue strand is DNA, and all of the rest of it, the pink part, is the other parts that would be in the nucleus. There are several steps in prophase. First, DNA coils tightly and becomes visible as chromosomes. So here's this long strand of DNA that we saw in the last slide. And what it coils around are protein molecules. And then these protein molecules, it would kind of be like if you took a piece of yarn and you started twisting it and twisting it and twisting it and twisting it, and what ends up happening is the yarn twists in on itself and continues to do that. But DNA does it in a very particular way. The second step in prophase is the membrane around the nucleus disappears. So up until this point, the DNA has been contained in that nucleus and in the nucleolus, and both of those membranes disappear. There are things called centrioles. A centriole is going to be used to actually pull the chromosomes, or pull the halves of the chromosomes apart. Those centrioles go to either end of the cell, called the poles, and the spindle begins to form. And last but not least, the job of the spindle is to attach to the middle of each chromosome and pull the chromatids, which, if you have a chromosome, half of it is called a chromatid, and those 2 halves are identical information. What would be the reason for having 2 identical halves of DNA information in one chromosome? Hopefully you guessed it was because when the cell splits, the chromosome information, the DNA information, the identical information needs to go into each new cell. So in the right half cell and the left half cell, if you think about them like that, need to have the same information that the original cell had. So the chromosomes, which are, there's 2 of them, and each chromosome has a partner, and that partner has the same information on it. So you don't need to know all of these terms, but just so you can kind of get an idea, this space, let's pretend this whole white space would be the nucleus. You can see there's no nuclear membrane anymore. And these X-shaped structures are the chromosomes, which are those coiled up pieces of DNA. This chromosome would match this chromosome. They would have the exact same structure, the exact same DNA information on them. And in the middle of each chromosome, you see these threads that go the whole way across the space that's left. And these threads are what pull apart the sister chromosomes. They pull them towards the centrioles, which are kind of like the key, or the grouping of all of those threads together. At the point in the center of every eukaryotic chromosome, it's labeled the kinetochore. Fibers attach, and those fibers are called the spindle fibers, in order to pull the 2 halves apart. In mitosis, the 2 halves of the chromosome don't get pulled apart. That happens in our next lesson, which is meiosis. Here you can see, this is called a karyotype. A karyotype is a picture of an individual's chromosomes. And you can see that each chromosome has a matching pair. They're matched by size, and in reality, the DNA in each matching pair is the same. We'll talk more about what DNA looks like and how it's structured in our next unit. The only 2 that don't match are these 2 at the end. This is the X chromosome and the Y chromosome. You may have heard of these before. The X and the Y control the sex characteristics for males and females. So if you have XY, 1 X chromosome and 1 Y chromosome, then you're male. And if you have 2 X chromosomes, then you're female. So backing up for just a second. Think of prophase as the beginning, pro as pre. This is before anything really starts to happen. The next step is metaphase. In metaphase, the spindle fibers from the centrioles attach to each chromosome. So here's where these fibers actually attach to the chromosome. And the cell gets ready to separate those chromosomes and pull them apart. But before they can separate, they have to align the chromosomes in the middle of the cell. If you had me for 7th grade, we did an activity where I took pairs of socks, and in the correct, for the group that did it correctly, the pairs of socks were already bundled up. So you had a left sock and a right sock, and those socks matched each other. And all you had to do to form 2 piles was pull the socks apart and put them down in 2 piles. That's what's happening in metaphase. Remember, meta is the middle. So first we had prophase, then we had meta is the middle. This is a picture of a cell going through metaphase. The chromosomes are the dark lines. They've lined up at the equator, or the middle of the cell. The asters have lined up at the poles. This is just where those spindle fibers are attaching to. And the spindle fibers, have attached to the chromosomes, and they're getting ready to kind of shrink and pull those chromosomes apart. So which cell in this picture is going through metaphase? Obviously, it's this middle cell here where the chromosomes have lined up. Those chromosomes need to line up in the middle so that you get the correct half of the chromosomes on one side and the other half on the other. If they didn't line up in the middle with their partners, then who knows which chromosomes would end up in which half of the new cell. The next step is anaphase. So we've had prophase, metaphase, anaphase. In anaphase, these chromosomes are actually pulled apart or separated. The spindle fibers shorten or shrink so that the chromosomes get pulled to the ends of the cell. Remember that ana is apart, pro is pre, meta is middle, ana apart. And this cell in this picture is going through anaphase. You can see they were lined up in the middle and now they've been pulled to the ends. Here's another cell going through anaphase. The sister chromatids, those duplicate chromosomes, are being separated. The next step is telophase. Here's where everything kind of gets finished. The chromosomes get completely separated, and in plants, something called a cell plate forms in between. In animals, it's called a cleavage furrow. And then the nucleus and the nucleolus reform around that DNA that has been pulled to the end. And then the chromosomes uncoil, they unwind. This is a plant cell, that's that plate, and here's the cleavage furrow in the animal cell. So in this step, all of the other stuff starts to get separated as well. And when all of that other stuff actually goes into each side of the new forming cells, it's called cytokinesis. This is the division of the cytoplasm and all the other organelles like the ribosomes and the mitochondria and the vacuoles and all of the rest. And it occurs after the chromosomes separate. So the chromosomes are the important things. They need to have the exact matching pair in each side, and then the rest of it kind of splits up based on where it's located. And when cytokinesis is complete, you have two identical daughter cells. So you can see the nucleus is forming around the chromosomes and they've begun to unwind, and then you have this plate in the middle. Here you see the M phase, or the mitosis phase, running all through. And it'll rerun again so I can describe. There's prophase, everything dissolves. Metaphase lines up in the middle. Anaphase pulls apart. Telophase splits. So part of your assignment today is to answer this question. Put the following in the correct order, and I can guarantee you that this question is going to be on the chapter test at the end of Unit 4, or a similar one. So you have pictures labeled A through G. You need to put them in the correct order using the letters underneath each picture. Just a couple of hints to remember. First, to remember the stages in order, remember PMAT. Remember pro is pre, meta is middle, ana is apart, and telo is when they split. If he needs to pause the presentation now to continue looking at these pictures, that's fine. If you have any questions, as always, email me. If you have any issues with submitting assignments, please let me know.